Well, good day to everyone joining us and welcome to today's X Talks webinar. Today's talk is entitled Enhancing AAV Full Capsid Enrichment with an Optimized AEX Platform, a case study. My name is Ryan Muse and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speaker. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using this same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Technova, who developed the content for this presentation. Technova makes solutions possible. Since 1996, Technova has been innovating the manufacture of critical reagents for the life sciences industry to accelerate the discovery and development of novel therapies that will help people live longer, healthier lives. They offer fully customizable solutions for every stage of the workflow, supporting industry leaders in cell and gene therapy, molecular diagnostics, and synthetic biology. Now, I would like to introduce our speaker for today's event. As Technova's Director of R&D, Bella Neufeld leads a gene therapy bioprocessing research program that develops products using viral delivery systems. She spearheaded the creation of Technova's first proprietary product line for AAV gene therapy development, AAV Tech Solutions, and successfully launched the AEX buffer screening kit. Neufeld's work focuses on resolving key bottlenecks in gene therapy workflow with ongoing development of upstream, downstream, and analytical solutions. But now, without further ado, I'd like to go ahead and hand the presentation over to our speaker for today, Bella Neufeld. You may begin when you're ready.
Thank you, Ryan, and thanks everyone for joining me on this webinar today. I'm thrilled to be here and talk about some of the work that our amazing R&D team has been doing at Technova and the collaboration we have with Via Sartorius, specifically, as Ryan mentioned, around enhancing AAV full capsid enrichment with an optimized AEX platform. Again, this is a case study with Via Sartorius. So before I jump into any of the data around this study, I do just want to introduce those of you who are not as familiar with Technova and the products that we make. As Ryan mentioned, we've been around for quite a long time, and we're really known for three categories of products. That is bacterial broth, media, agar plates, and buffers and reagents. And so one can imagine, given that type of product, we serve many of the different life sciences and have many different SKUs or products associated with those. We're really known for smaller quantities, so less than 1,000 liter manufacturing scales, and our ability to go from RUO to GMP grade. So we go with you from product development to all the way to clinical manufacturing. And as I mentioned, we're known for really our custom reagents. And so in this space, while we serve a lot of the life sciences, we really fit well into this area of cell and gene therapy, but really specifically gene therapy for our ability, again, to do custom manufacturing at very discrete concentrations. So one of the themes you'll notice in this presentation today is the need for highly custom, highly discrete values for each of our buffers that we manufacture within a gene therapy landscape. As I mentioned, we've been around for quite a long time. Our first 20 years of really existing was building our modular manufacturing platform for our ability to make these many, many different types of products and SKUs. But it was really around 2017 and even a little bit later that we decided to put some significant investment into the company, both from a commercial and marketing standpoint, but also why I'm here today for our R&D lab. So we've been around for just about 3 years, almost 3 years. And by establishing an R&D lab, we've really been able to grow both our expertise in the space, but also specifically around AAV. And so from here on out, now we're really in the scaling phase. But again, for these last 3 years, we've really built out our core R&D team and the end-to-end AAV platform in which we'll be discussing today. And so in part of being able to do this, of having this amazing R&D lab and team, we've been able to partner with some really phenomenal companies such as Sartorius Via to really understand how our processes can fit together to help streamline some of these AAV gene therapy purification processes of what we think of as one of the major bottlenecks, specifically around full capsid enrichment. When we decided to start an R&D lab at Technova, we quickly understood the importance of finding all the gaps within the end-to-end AAV workflow. And so we knew very early on that we were going to have to build out this capability ourselves in-house in Hollister, California, where our headquarters is. And so we did just that. We built an end-to-end platform all the way far upstream in plasmid production and development, where we do all our own cloning and plasmid design, all the way down to the final formulation step. And one can imagine, for a buffer and reagent company, we spend quite a lot of time thinking about the downstream purification part of AAV as the buffer component becomes so incredibly critical. So while I'm sure you're all very familiar with the sort of end-to-end workflow that I'm showing here, the key point is that we've had to establish this in-house at Technova so that we could understand better what these pain points are. And when we did that, when we really dug in and started to make our own AAV and purify it and run the analytics, we found that there was one particular area that we should focus on first. And while we have lots of solutions for the entire work stream, we did find that within the chromatography step, this concept of having discrete buffers that both are at the exact ratios and concentrations that one would expect, and the ability to scale in a reproducible fashion all the way to GMP, seemed to be incredibly critical at this stage. So this is where we wanted to focus our original efforts, and this is ultimately what ended up launching, as Ryan mentioned, our first AEX buffer screening kit, our first proprietary product out of Technova. This is a deeper schematic of what we think of as really the downstream purification. So all the way from really your endonuclease treatment, when you're trying to chew up your residual DNA, all the way down to your final formulation. Again, within that chromatography step, we generally think of two different aspects of this. One could be the affinity, where we're doing more of a capture step, of which we also have some novel solutions to help with this particular step in the workflow. We really wanted to focus on what we consider to be a significant challenge in the space, and that is around full capsid enrichment, or the polishing step. And this is where, we come at this from an ion exchange perspective, not really thinking about as much as the centrifugation workflow, again, because we're interested in those who are really looking to scale their processes. So we're really focused within the chromatography stage, and within ion exchange, really trying to understand how these buffers and the components within the buffer might affect our ability to do full capsid enrichment, specifically from an anion exchange perspective. And this is where our partnership with BIA really came to fruition, to say, we've developed these highly customized, highly robust buffers with all of these discrete parameters, be it pH, elution, salt, etc. And they really have a standardized platform using their monolith technology that allows really high throughput of these types of processes that we're trying to probe. So we quickly wanted to partner with them to see how robust can we make this process, and how much can we really push to get as much full capsid enrichment as we can, while of course trying to maintain as much recovery as possible. So this is a bit of a layout of what we're going to be discussing today. Again, in our landscape, we really wanted to identify what was a critical pain point within the downstream process workflow that we could theoretically address in given our expertise in buffer manufacturing from both the discrete standpoint and the ability to scale. What are those critical components within a buffer that drive this? And of course, what fell out of this was a really wonderful case study that we did with BIA Sartorius. And then ultimately taking that data, or that concept that we discovered with them, and expanding it even further to really develop an off-the-shelf buffer screening kit that anyone could use given their serotype. And that's really what we're going to be discussing today, is how did we get from this pain point that we identified in the field all the way to an off-the-shelf buffer screening kit with a wonderful case study in between. For those of you who are not as familiar with the concept of using ion exchange to do full capsid enrichment, that's what this schematic is trying to show here. So essentially what we are doing is exploiting very small changes in a PI of the outside of a capsid, given whether that capsid contains or does not contain the transgene, so whether it is considered empty or full. And we can exploit these small differences either using cation exchange or anion exchange. For the remainder of this talk, and all of the work we've done currently at Technova is all around the anion exchange step. But in order to do that, in order to really drive those differences, as one can imagine, they are slight, we quickly recognize that what is going to make a big impact is what is contained within that buffer. So both the equilibration and the elution buffer in which you employ to do this step, to do this anion exchange step. And we took a large screening list of all of the things that we thought might have some impact, because quite honestly, we weren't sure when we started out on this study. So it could be something as simple as the base buffer, but also at what concentration. The elution salt, of which we've screened many different elution salts, 10 actually. The acidity or pH of that equilibration and elution buffer. And then when it comes to things like excipients, surfactants, and stabilizers, not really knowing what the intended effect would be, but seeing if there was one. Not just saying, are they present, but to what degree are they present, and how might that impact its ability to get stronger recovery, stronger purity. And so all of these parameters, essentially, when we addressed them and looked at them, the goal, right, is to find some sort of idealized, optimized equilibration elution buffer that one could apply towards their system and get out exactly what they're looking for, whether it be recovery, purity, or both. So enter the study that we did with Sartorius via, where we essentially employed a design of experiments using all of those parameters that we just discussed, base buffer, elution salt, pH, etc. And that was really the piece where Technova came in to build this robust set of buffers. We started with 10 as the buffer set, right? So really 20 equilibration elution. Pass those off to BIA, where they're employing their Simultas QA1 monolith column. And they ran it on an AAV8 serotype construct, looked at a variety of evaluation methods, and from that, we were trying to get out so that if we run this type of study, what would the optimized buffer component be, and could we scale it? So for their evaluation methods, of course, recovery by ddPCR, purity by their PAT-fix system, in which we employ a SIMAC empty full column, 260-280 ratio, the resolution of the peaks, and then the retention on the column, so the conductivity. And that was the crux of the study. Now, just a couple of more considerations for how this was actually carried out. As I mentioned, we had 10 buffer sets to start. This material was processed using another monolith column that was just upstream of the AEX. So in this case, it's an SO3 CEX purification step. The incoming material started with 46% full capsid, so that's the load material for the AEX, and that's based off HPLC. That material was diluted 40x to get into the correct conductivity to actually be able to load it effectively on the column. And then fractions were collected based off the switching of the A260-280. So the final piece here to keep in mind is what we were looking for when we plugged this into a DOE model, of which there were actually 2 DOE programs that we used to assess this, is we wanted to see at least 80% recovery and at least 85% full capsids or purity as the output of this DOE. I'm not going to run through all 10 chromatograms that were used in this study or that were elucidated, but what I will show you here is that this is just 2 of the 10, and this gives insight. So the one on the left, you can see we get very little peak separation at all, and very little resolution between the empty and full. We just get 1 peak. Where on the right, you can see we're already starting to tease apart that empty and full with the initial 10 buffer set. So already by choosing the parameters that we chose, again, those initial set that we defined, whether it be the base buffer, the excipient, the surfactant, within that 10 set, we're already seeing some really solid candidates that could potentially work for your own scale up or transition to the next step. But once we ran all of these, we put them back into a DOE, and we came out with what to be the optimized ultimate equilibration and elution buffer. And once we figured out what that was, of course, we wanted to scale it up at Technova, send it back to Sartorius so they could run it again on an AAV and do multiple cycles, and again, confirm that the output of the DOE was what we expected. And the point of this was to really say, is this a reasonable or rational approach to be able to determine what the components of the buffer should be to really get what we are looking for? Again, that high recovery, high purity. And indeed, we were very pleased with the results to find that yes, by using this approach and seeing the output of this DOE, the optimal buffers were indeed 85% full AAV with 80 to 95% recoveries, depending on the run that was conducted. So the chromatogram here shown is one of the 2 runs that were used with the optimal buffer. And the table is really showing, based off the predicted, again, from 2 different DOE programs, MODI and JUMP, how that compared to what the actual experimental output was. And I think the key here is, number one, it's really critical to us that we are able to get significant resolution between the empty and full peaks, such that if someone wanted to scale, not using a linear gradient, that's the other piece here, is that they are all linear gradients at this stage, that they would be able to apply some kind of isocratic hold, whether it be step elution or an isocratic plus a linear. But if you don't have strong resolution between your empty and full peaks, it's very challenging to understand how to implement that type of isocratic hold. So that is one piece. And then, of course, the second and third are really around this purity and recovery. So again, if we think about purity using their HPLC method here, we're getting very, very high purity in that full peak, and significantly good recovery based off ddPCR measurements. So this was one of the case studies that we ran with VIA Sartorius. We're very excited to share with you today, and we hope this approach can be used across many serotypes, not just for AAV8, to help get at what that optimized buffer parameter might be for you. So before we move on to the next stage of the process, we do have a poll for the audience.
Yes, indeed. It's appearing on everyone's screen at this time. People can participate by selecting on any of the answers that you see in front of you and then click submit. The question that we're asking is, what serotypes do you work with? 2, 6, 8, 9, or custom? We'll give everyone some time to consider their answer again to the question of what serotypes do you work with? Looks like most of you have submitted your answers, so thank you very much for participating. Let's look at where your results have come in. It looks like we have ourselves 33% of you selecting custom serotypes, then 22% each for 9 and 6, and then 11% for 8 or 2. So thank you very much for participating again, and it's now back to you.
Well, thank you for participating. It's really helpful for us as we're designing these buffer screening kits to get a sense of what people are working on and where their pain points are. So not a big surprise, quite a big spread, a lot in the custom bucket, so appreciate the answers there. So while we address that this was indeed a rational approach to take, is to say, let's screen, let's do a DOE on these types of parameters and see how strong the output can be given this type of screen that we did. We wanted to take it really to the next level to say, okay, that was one case study, one starting material, right? One process that was put in place, and we were able to achieve a pretty solid recovery and purity profile given this approach. But how robust can we make this? How much can we really push the envelope if we do hundreds of AEX runs and try to tease apart what those parameters might be, given all of the different changes that we know each of our clients might face? And that's really what we're showing in this bottom table is, what if your upstream platform is significantly different? What if you have different media, different transfection agents, different harvest conditions? Likewise, what if your downstream platform is different? And so each of these, when we think about the upstream and the downstream, and then of course the transgene effects, we know that your starting material might have a significantly different amount of purity, percent full to start with. And so how robust can we make this process given different starting parameters or different starting material? We also wanted to play around with different AEX platforms and see how robust the screening kit is for that. And then finally, we added an additional critical quality attribute, or CQA, in this step that's really centered around this infectivity measurement. So what we've found, and we hope we have convinced the public of, is that it is incredibly important to understand the in vitro conditions in which your viral capsid exists, given all of this process parameter steps that we're working on. So for example, we have seen that the buffer components do make a difference. Whether you're testing ELISA or PCR HPLC, they have an effect on the ability to have infectious titers because of the actual matrix of the buffer itself. So having that additional point to say, and our infectivity is significantly strong or high compared to other process parameters, was also very important. And so the next data set I'm going to show is just really showing exactly that. How far can we push this approach, given that we wanted to change a lot of parameters before we get to the AEX step? So once we went from really the VIA Sartorius collaboration, we developed our own AEX buffer screening kit for AAV8, and we vetted it on a lot of different platforms. This is just one example of that. So Significantly changing our upstream, this is an example of what the affinity capture step looks like, which was done just prior to the AEX polishing step of applying our buffer screening kit. And what you can see here is that, unsurprisingly, depending on your upstream, you're going to get significantly different percent fulls in that affinity capture step. And of course, the profiles will then look different accordingly. However, as we move to the AEX and we're able to achieve that full capsid enrichment, we can still get significantly high percent full using our buffer screening kit. And indeed, the chromatograms could look substantially different. So on the right-hand side, we have the AEX polishing step of the same two upstreams. And in one case, you can see there's significantly more empty. And in the other case, you can see there's quite a bit less empty. Again, we suspect that's based off the upstream parameters that we put in place. But the point here is that when you use our buffer screening kit, it shouldn't matter what your upstream platform is and what your incoming percent full is. You can still achieve significant enrichment and recovery using this platform. And we had a similar story when we were looking across transgenes. So things from GFP to an IL-12, again, significantly different starting material, one as low as 13% for a variety of reasons that we really tried to push this robustness and see how well this could work on a completely different platform. And again, if we take that affinity, right, profiles look completely different, and we take them to the AEX polishing step using our buffer screening kit, we can also achieve rather strong full capsid enrichment and also strong recovery. So this was very promising to us that we had established this AEX buffer screening kit that could be applied across different upstream, downstream platforms. The last piece here that we really wanted to encourage everyone to think about is then how do we actually scale? This is something we think about at Teknova a lot because our whole manufacturing platform is based off going from RUO to GMP, and we help our customers through that process. And so how would that be applied to something like the AEX buffer screening kit? Well, in this case, again, this is coming back to that idea of, do you have good resolution? Do you have strong conductivity differences between your empty and full peaks? And if you do, can you exploit that for some level of isocratic hold or step elution? And that's exactly what we're showing here. So using the buffer screening kit, we've identified where those conductivities lie. We've built additional buffers to be able to tease that apart and really extend this to show if you didn't want to do a gradient and you wanted to then switch over to a step elution or some kind of isocratic hold, could we help you in that process? And indeed, this is showing that we can. So given that, we have another poll question.
Yes, thank you. So appearing on everyone's screen is our second poll question. This time we're asking, when you scale your chromatography step, which method do you utilize? Your answer options are linear elution, step elution, or a combination of linear and isocratic elution. We'll give everyone some time to consider their answer. As with the previous one, please select one of the answers you see in front of you and then click submit in order to participate. The question again is, when you scale your chromatography step, which method do you utilize? Looks like most of you have your answer in, so thank you so much for participating. Let's look at your results. Here we have 50% of you selecting step elution, 40% for a combination, and then 10% coming in for linear elution. Thank you again, and it's back to you, Bella.
Yes, thank you. That's really helpful to get a sense of from the audience how you scale. And that's exactly what we were thinking was likely the case, that if we do these sort of linear gradients in the process development stage when we're screening lots of conditions, in this case buffers, trying to understand which one works best, how then do we take that buffer and scale it all the way using a different modality? And so hopefully we will be here to help with that process. So I've sort of given you an outline of how we developed these kits, these off-the-shelf buffer screening kits. Everything you've seen today has really been around AAV8, but I will say we also have one commercially available today that is for AAV2. We continue to work through the serotypes, hence why I was curious what you all were working on in terms of your serotypes, and to continue to create these buffer screening kits that will be off the shelf that are serotype specific. And with that, of course, I would love to thank my amazing team for doing the wonderful work that they do every day to help us develop these. And of course, our partners at Sartorius Via, and all of you for taking the time to listen to me today. I'm happy to take any questions that might be up.
Well, thank you very much for this very insightful presentation. At this time, I'd like to invite the audience to continue to send their questions or comments right now using the questions window for this Q&A portion of our webinar. I've already received some questions for you, so we'll get ourselves started with those. The very first question would just like to know, how does the buffer kit work on custom capsids?
So great question. So we actually don't have access to any custom capsids. We only have those that are commercially available off the shelf. And so while we try to do as much tweaking as we can between serotypes and between capsids to see the robustness, we don't have a lot of opportunity to try that. We are pretty hopeful though, because we've already sold a number of these kits to people that are working with, whether it be AAV2 or AAV8. They are considerably modified capsids based off that, and the kits have worked really well for them. So we're hopeful that this is going to be working across multiple serotypes, including custom capsids.
All right, wonderful. Thank you so much. The next question here is wondering what serotypes are coming next. Further, will there be a universal screening kit that covers any capsid?
I think we would all like a universal screening kit, and I think we will get there. I think the more studies that we do like this, the more screens we can do, the more overlap we find between buffers. And it's not necessarily even just the buffer formulation, but it's also what parameters make the biggest impact on the ability for full capsid enrichment. So what's that factor contribution? That's the output of the DOE. So the next screening kits that we intend to have are 6, 9, and 5. So stay tuned for those serotypes. I'm very keen to hear if people have struggles with particular ones. So please reach out. I'd love to know. And I think once we go through these, as we start to diversify the kits and the serotypes that we test, I do suspect we will ultimately have a more universal buffer screening kit that one could buy and use across their constructs. So stay tuned.
All right, good to know. Thank you. I've got a question here wondering which surfactant and stabilizer was used in the AAV8 equilibrium and elution buffer.
So we don't actually give out the formulations for these buffers in the screening kits themselves. It's nothing incredibly novel or different than what you're probably all working on, but the formulations of the screening kits themselves are proprietary to Teknova for the very fact that we don't want them to be recreated in your house. So we hope that you buy them from us. Now, there are ways to get access to the formulations if you do partner with us and want to scale through GMP. So again, if you're interested, please reach out. But for the screening kits themselves, we don't give insight into what the actual components are.
All right, very good, thank you. Another question here is wondering why a kit of multiple buffers instead of just one buffer per serotype?
So the output of the DOE obviously gives many different parameters that might be ideal for your situation. So one reason that we don't have just one buffer is what I mentioned before, in the fact that we know that everyone has their different platform. So they might have a completely different upstream, a completely different downstream, different analytics, and all of that will affect really which buffer works best. But even more than that, we find that there are some buffers that actually are better for high recovery, and some buffers that are better for high purity, and some that are the best for infectivity. So depending on your end application and your workflow, one buffer might be more suitable for your actual end goal, whether it be really high recovery, like you need the most titer you can possibly get, or you don't need as much titer, but you need really, really pure samples, in which case each buffer might be specific to that.
All right, wonderful. Thank you so much for that answer and for all of the answers today. However, we have reached the end of the Q&A portion of our webinar. Now, if we couldn't attend to your questions, know that the team at Teknova will follow up with you, or if you have further questions, you can direct them to the contact information on your screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Now, I've also sent you a link in the chat box, and with this link, you'll be able to view the recording of this event on this page, and you can also share this link with your colleagues when they register for the recording here as well. So I encourage you to do that. Now, please join me in thanking our speaker for her wonderful time here today. We hope everyone found the webinar informative. Have a great day, and thank you for coming.
Thank you.